Being that the vacuole is a large, membrane-enclosed space within a cell filled with a variety of materials, its form fits its function in that it provides a space to hold materials and waste as well as maintaining pressure within a cell. If the vacuole was whole and did not have a free space within itself, it could not store any materials. The evolutionary significance of vacuoles is that without such organelle, cells would be extremely limited in their ability to maintain their internal environment. Because the vacuole stores nutrients, waste, and controls pressure, without the evolutionary benefit of having a vacuole, cells would have less ability to protect themselves and maintain their internal environment. The vacuole participates in a variety of cellular functions, including isolating materials that may be harmful to a cell, containing waste products, containing water, maintaining internal pressure, and the structure of a cell.
This is the Golgi apparatus. It receives proteins and lipids from the endoplasmic reticulum and sorts, modifies, and synthesizes the fats and proteins it receives. It is composed of membrane-bound structures called cisterna, and each cisterna possesses special enzymes which help to modify and synthesize and transport proteins and lipids. Every cisterna bounded together forms a membrane and separates the apparatus from the cytoplasm. The Golgi apparatus evolved early in eukaryotic cells and was a significant step in the prokaryotic versus eukaryotic transition. Its job of synthesizing fats and proteins helped to distinguish eukaryotes from prokaryotes and create more complex cells. The process the Golgi apparatus takes part in, as described earlier, is in the synthesis and packaging of proteins and lipids, which is very vital to the cell's well-being.
This is the nuclear envelope. So as you can see here, it is surrounding the nucleus inside the cell. So the first thing it talks about the form fits function. So it does have a double membrane, and the double membrane is actually connected. So the inner part protects the nucleus, while the outer part is connected with the endoplasmic reticulum, and that allows transfer of it turns the mRNA into proteins and transfer between the nucleus and the endoplasmic reticulum. Also, it surrounds the cell, and therefore that's the best way to protect the nucleus. And finally, it also has pores that allows things to exit and enter. So the pores kind of facilitate and what exits and enters into the nucleus. In terms of evolutionary significance, it provides protection for the nucleus as the membrane provides for the cell, and also it's thought to have been descended from the first ancestors who had nucleuses. And processes, it protects the nucleus and also regulates what goes in and out of the materials into the nucleus. So this is the endoplasmic reticulum. As you can see here, it is right underneath the nucleus, nuclear envelope, in the cell. So it is connected to, it's all connected, so it's all one huge lumen, and and this allows it to, and it's all really long. So this allows it to ship materials to many different organelles around the cell, because it's so long and it covers so much space. In terms of evolutionary significance, it evolved into two, so the smooth and the rough. The rough does more like manufacturing of lipids, and the rough has ribosomes on the outside, which assemble the proteins. So two kind of parts of that. And then also in terms of processes, it's involved in the protein creation and processing and sorting, and also it ships proteins around the cell, and it ships many other different materials around the cell to different organelles in order to keep the entire cell working.
Chloroplasts are a double-membranet organelle that is the primary site of photosynthesis inside of the cell. Inside, the chloroplast is filled with stroma, a colorless fluid, and grana, which are stacks of thylakoids. Thylakoids are where the light-dependent reactions for photosynthesis take place. The organelle is believed to have originated from cyanobacteria through endosymbiosis from eukaryotic cells. The organelle itself plays a large part in photosynthesis, which is essential to the release of oxygen for humans to breathe and for the plants to create ADP for themselves. The mitochondria are a double membrane-bound organelle in the cell. It is the site of aerobic respiration and the generation of ATP, adenosine triphosphate, the cell's primary source of chemical energy. It also helps control cell growth in the cell cycle. Similar to chloroplasts, the scientists believe that mitochondria were originally aerobic prokaryotes before being engulfed through endosymbiosis. Mitochondria are made up of a multitude of parts such as the inner membrane space, the cristae, and the matrix. The cristae, which are located in the membrane, expands the surface of the inner membrane, enhancing its ability to make ATP. The matrix is where hundreds of enzymes are located, as well as the mitochondria's own independent DNA and ribosomes. Molecular machine that produces proteins for the cell through translation. In translation, the ribosome reads mRNA and determines the correct sequence of amino acids to form the protein. The ribosome is made up of a large and small subunit which bind the various parts of proteins together. Scientists believe that before ribosomes were created, proteins were made by ribozymes, a hybrid of ribosomes. At some point, it separated into its two parts, RNA and a ribosome, which translated RNA into proteins.
Being that the cytoskeleton structure is composed of a framework of protein fibers, its form fits its function in that the web of protein fibers can extend to hold on to the organelles of the cell. Because the cytoskeleton's main role is to control organelle movement, its network of fibers allows it to do so. The evolutionary significance of the cytoskeleton is that without such, the cell would not be able to function nearly as efficiently. Without this framework of pathways of which holds the organelles into place, as well as allows for the movement of the cell would not be able to perform basic functions needed for survival as efficiently. Therefore, without the cytoskeleton, the cell would be less equipped to function as a unit. The cytoskeleton participates in the maintenance of cellular structure, transportation of the cell, and movement of cellular organelles.
Hello everybody. In this video we will be talking about the lysosome. The lysosome is a vesicle containing enzymes that help break down worn down organelles and bacteria attained through phagocytosis. It starts off with enzymes formed in the endoplasmic reticulum that are known as acid hydrolases. Such examples of acid hydrolases include proteases and lipases. These enzymes are packaged in the Golgi body, where a single outer membrane that constitutes of phospholipid bilayer buds off and forms a vesicle. This is the primary lysosome in this stage. The form of the single outer membrane constituting of the phospholipid bilayer allows for fusion with other vesicles, such as those containing bacteria and worn down organelles. Once they fuse, it is known as a secondary lysosome. In the secondary lysosome, the inner environment is at an acidic pH in contrast to the cytosol around it, which is at a pH of 7.2. Now, its evolutionary significance, much like that of the protobionts, it allows for a separate environment in which digestive processes can occur separate from that of its outer environment.